Well, thank you so much for inviting me to be here and present some of the work we've done in the lab to Professor Yu's challenge about maybe what we can do for multi-omics AI. So our other panelists as well have also highlighted some of the other challenges, but I'd perhaps like to begin just by briefly reiterating some of these challenges, in particular from a computational standpoint. I think the first thing I want to just highlight is the dimensionality of omics data sets and different omics layers, especially compared to a lot of other data modalities more frequently seen both in medicine, but also in other AI applications. These can range from kind of tens of thousands of different variables to hundreds of thousands and even millions. Another couple of challenges that are particularly common in multi-omics data sets with like where computational methods can really struggle are limited labeled data, where we're then more likely to pick up on very spurious relationships between our variables and our outcomes of interest, as well as interfeature correlation, which can confound the learning process. So just to briefly explain what I mean by this, let's say we have a gene X1 that when it's really highly expressed might cause some phenotype Y, but also causes gene X2 to be highly expressed. Kind of the feature we want to identify or we want our predictive model to use really is this gene X1, since it's causing our phenotype Y, but the presence of variable X2 makes this much more challenging. So I'm going to begin by talking about a method that's been applied for polygenic risk scoring. And this first approach is a method developed in the lab by former PhD student Jinsun Yoon, as well as others in the lab and Professor Van der Schaar, called VIME. And this introduces a novel self-supervised approach for tabular data such as omics that allows us to learn from unlabeled data effectively to then be able to perform supervised tasks downstream. So to very briefly explain how VIME does this, I'd just like to consider the left-hand side of this figure on the slide. So we first take an unlabeled sample, and then we can corrupt this sample by masking out some of the entries at random and replacing them with other values that this variable could take, but that might not make sense in the context of the other values for that sample. And we then train an encoder network to be able to both predict which of these variables were masked and what the true underlying feature value should be. And this allows this encoder network to really understand the raw data on a deeper level. And then with what limited labeled data we do have, we can then train a predictive model on top of this labeled data in either a supervised or a semi-supervised manner. So as an illustrative application, VIME was applied to genome-wide polygenic risk scoring using data from the UK Biobank. And the goal here was to predict 6 different blood cell traits from single nucleotide polymorphisms using UK Biobank data. And VIME outperformed all of the benchmark approaches tested across all of these 6 different traits. A very different challenge that Andres, in particular, Professor Floto identified was that of biomarker discovery. And so in standard prediction problems, we often have a set of features or variables, and we want to train a predictor to maximize its predictive power. In contrast, in the case of kind of biomarker discovery, we actually want to identify just a smaller subset of the variables or features that are going to be the most predictive. And this then becomes a joint optimization problem of selecting kind of as few features as possible while still maximizing the predictive power of our predictive model. And there are several reasons we might want to do this. And I think the most pertinent for multi-omics data is really to get this deeper insight into our data and understand exactly which biomarkers are driving the phenotypes that we're interested in. But there are also, in maybe other domains, in particular issues around cost, but also around generalization. And we want more robustness to these nuisance features to overcome this challenge I mentioned earlier about this interfeature correlation. So to address this, we proposed a novel feature selection method called CEFS, which is joint work between myself and a former PhD student from the lab, Chang-Hee Lee, as well as Professor Van der Schaar. And to enable more accurate discovery of new biomarkers, we use this two-stage approach for our method. In the first, we follow a similar procedure that I described for VIME and pre-train this encoder network in a self-supervised manner to learn representations that are favorable for feature selection. And then we use this pre-trained encoder in order to select features that are most predictive of our target outcome. The key technical innovation here that really helped us was to handle highly correlated features in particular is how we selected this is how we selected the features and to really incorporate the correlation structure of the underlying data much more effectively than was done in previous approaches, which chose to ignore this. And I'm willing to take maybe some more questions about this if it's interesting in the Q&A later. We validated our approach on both transcriptomics data for peripheral blood mononuclear cells, as well as proteomics data from the Cancer Cell Line Encyclopedia, and found that our method CEFS outperformed a large number of other feature selection approaches in both cases. And additionally, we are frequently able to identify and validate the discovered features, with many of the top-ranked features shown on the right in blue for the proteomics dataset only discovered with our self-supervised approach. Here we have, here in that work I just described, we might have a subset of selected features. But a question we can ask is there further structure here we can discover? Often, let's say it's not just one mutation that is driving a disease, but maybe it's the occurrence of two mutations jointly. But these previous approaches for feature selection won't be able to identify this. They'll just identify a set of relevant features. So a natural way of thinking about this additional structure is, instead of just one big list, discovering what we're going to call composite features, which is effectively groups of variables that have some form of joint importance with each other, but that are distinct from these different groups. And there are many scenarios where this plays out, both within multi-omics datasets and beyond. And so to address this, we proposed a new machine learning model called CompFS for composite feature selection that's composed of an ensemble of feature selection models, similar to the ones I just described earlier, as well as an aggregate predictor. And this enables us to identify these different groups of predictive variables. Finally, I want to turn our attention to multi-omics integration, which is one of the most significant challenges that exists in this area. And one of the challenges that Professor Foto identified in particular in his presentation was how to integrate and learn from multi-omics data, and in particular when we have missing omics observations and various missingness patterns across different samples. And so conventional solutions for this often discard samples with missing omics, which loses samples. Alternatively, they could discard different omics layers which have missing samples, but this loses omics-specific information. Or alternatively, they're forced to impute missing omics observations, which may cause data distortion. To address this, former PhD student from our lab, Chang-Hee Lee, together with Professor Van der Schaar, proposed a novel approach to solve this problem called Deep IMV that's able to integrate data from multiple omics layers with arbitrary missing patterns, both during training and at prediction. And it achieves this via a set of view-specific encoders and using an informational bottleneck approach. This approach was validated using data from the Cancer Genome Atlas, predicting 1-year mortality based on multi-omics observations, outperforming all approaches, including the popular MOFA approach, across different number of views, and in particular when learning from incomplete data that's more representative of real-world scenarios. As one final topic, Andres mentioned in his talk that one key development is to use omics to guide treatments. And to move in this much more causal direction, we need to understand both conditional dependencies and independencies between variables in the presence of treatment. But this becomes particularly challenging for high-dimensional data. So for this, former student Alexis Bella and Professor Van der Schaar proposed this new approach based on generative adversarial networks for performing conditional independent tests, in particular for this high-dimensional data. And their approach was validated on the Cancer Cell Line Encyclopedia to distinguish between genetic mutations that influence drug responses. And so for many mutations, as shown in this table here, actually the method GCIT agreed with a lot of the existing approaches and the literature. But in several cases, they noticed this disagreement between the newly proposed existing approaches, the existing computational approaches. But they found that the GCIT kind of decision about the conditional independences agreed with the literature, which I think really demonstrates the promise of this approach.